Hi, everybody. Welcome to today's webinar. Today, we are celebrating the July 2025 focus issue of Plant Physiology, and the topic is Numeracy, Realism, and Relevance, which is, I think, a very intriguing topic. We've got four speakers and a moderator. I'll be introducing the moderator, Merritt, but she will then introduce each of the speakers in turn. Before we get started, this focus issue is online now. I'll put the link to it into the chat. This was edited by Jen Brophy, Yang Rong Lin, Jackie Shanks, Allison Smith, myself, and Andrew Hansen. We will be posting the recording of this webinar to YouTube along with all the other plant physiology and plant cell recordings. And if you enjoy listening to webinars, you can find a whole bunch of great topics there. We would like to thank the ASPB for allowing us to host these webinar series. It's supported by ASPB members. If you are not a member and you'd like to become one, you can use the code PRESENTS10 and get 10% off your membership dues. The other thing I would like to mention is that starting next week, we have our annual plant biology meeting. It starts on Saturday, the 26th of July. But you can still register if you'd like to join us. It's in Milwaukee, Wisconsin. We have a questions box that you can type questions for the speakers into. Email me if you have any problems. And I will turn it over to Merritt. We're delighted to have Merritt serving as the moderator. She's an artificial intelligence and breeding research scientist at Corteva Agriscience. She got a BS in biology from the University of Hawaii and a master's from South Dakota State University. She got her PhD in plant genetics from Cornell University, working with Ed Buckler. And during that time, she investigated biological patterns that control pleiotropy and the regulatory impact of transposable elements on gene expression in maize. She also led a very interesting perspective paper that some of you may have seen discussing with an international multidisciplinary team that advocated for rigorous field-based trials when evaluating genetic improvements on crop yield. And you'll hear more about that during this hour. To ensure that discoveries actually translate into real-world on-farm yield gains. And at Corteva, she develops computational and AI-driven approaches to advance crop improvement through gene editing. So I'll turn it over to Merritt. Thank you very much.
Thanks, Mary, and thank you to all the panelists for joining us today. To get us started and kind of in the mindset of numeracy, realism, and relevance in plant science, I kind of want to turn it over to Jonathan first to talk about kind of what this special issue is about and where it came from. So Jonathan, go ahead.
Okay, thank you very much, Merritt. That's really nice to be here. I'm the lucky one because it's lunchtime here in the UK, and everybody else is either should be in bed or having their very last meal of the day. But it's great to be here. And the first thing that I really want to say is, a real thanks to Andrew Hansen for organizing this focus issue, and also there was an associated NSF workshop earlier this year. Andrew has been really sort of instrumental in driving the recognition of the issues that we're going to talk about this morning, or this afternoon, or this evening, wherever you are today, because I think both him and I sort of recognized that there were some issues associated with a lack of realism when it came to especially some of the more fundamental plant sciences. Basically, I think we were all concerned that there was a degree of over-egging what could, and the emphasis being on the word could, be achieved in the future, and we needed somehow to come up with some sort of almost intellectual framework that would allow us as a community to be more realistic and to be able to better serve the needs of society. And what Andrew was really great at is actually, basically putting that into a framework that it involves some practical ideas and concepts, whereas I was probably much more just sort of felt like I was tilting at windmills sort of things. And so I think it's again, I'm just saying, I think Andrew's done a really good job at just trying to frame some of our thinking around this. And I just want to share one slide that from my perspective, I think is relevant, because it really framed my thinking around this whole topic. And it was that I went to an EU conference 10 years ago, and I went as I'm a card carrying plant biotechnologist, and I'm interested in real world science, but I'm also working at the molecular level, at the biotechnological level. And there was a talk from a US professor, Philip Hardy, who was at the University of Minnesota, and he was talking about the timescales that it takes for something to have impact. And most of his examples were biotech examples, but actually in reality, you could other of his examples would have been the same using conventional plant breeding. And what was like the sort of scales falling from my eyes was I realized just how incredibly long it takes for something to go from being an idea, or even just actually you could have moved beyond the idea phase, and you could be you could have discovered something. And to go from a discovery to something that is actually a real world thing that you can grow in the field, buy in the shops, whatever, put in your mouth to eat. It takes, it's a decadal timescale. And so, it's not weeks or months or even years. And the challenge that we as a community have is trying to make sure that the reality of those timescales is recognized by our funders, by our policymakers, by our peers, and that we don't inadvertently or advertently try and suggest that the timescale for something arriving is going to be much quicker than it really is, because we're all under pressure. We're all under pressure to achieve, and we're under pressure to deliver. And so the temptation is to I think to suggest that positive outcomes will arrive more quickly than they will. Now, and I think this is a particular issue within academia, because I think if you're working in industry, there are a whole load of checks and balances to ensure that you're not going to sort of that you know where you are on a pathway to delivery. Where in academia, I think we have a degree of freedom that allows us to be maybe slightly more to be more optimistic or more vague about how we will deliver impact. And so to me, this whole focus issue is really about trying to think around those topics, and that and just understand what it takes to go from a finding you've got a useful thing in a transgenic plant in the lab to actually how long would it take to get to a product in the field or in the supermarket. How long would it take? What's the pathways to go through? And I'll talk about that in a minute. But that's really, and it's really, to me, it's all about how do we help build a community that understands the issues, and how can we radiate that information out to the wider world, rather than suggesting that this time next week we're going to have GM whatever, or a new trait or whatever, and it's and we'll be feeding the world, and all those phrases that we're all guilty of using, but in reality probably are becoming slightly overused, if not slightly trite. So I'm going to stop now, because I think we can then move on to some more actual concrete things. But that's my pitch about why I think all of this stuff is interesting, important, and because it just takes a long time. And so you got to start. And the sooner you start, the sooner you'll get to the end, which is a good point for me stopping.
Awesome. Thank you, Jonathan. And just to give a feel to everyone on the call about how this panel is structured. So each of the speakers I'll introduce, and then they're going to talk for roughly like 5 minutes about the paper that they have in this special edition. And then I have a few pre-prepared questions for each of them, and then the entire panel. But then as folks are talking or answering some of those questions, feel free to put your questions in the Q&A if it's directed towards a specific speaker or a specific paper. Feel free to write their name along with your questions, or just post any question that you have in that Q&A. And the second half of the panel is more for all of your questions. So we'll get to them when we can. So the first speaker on my list is Sam. So Sam is currently a PhD student in Ron Millow's group at the Weizmann Institute of Science. Sam completed his undergrad and master's degree at the University of Oxford in biochemistry with a focus on bioinformatics and how plant genome editing can be used in agriculture to help mitigate climate change. His current research focuses on quantifying the environmental impact and economic implications of new methods of food production and their role in limitations in future sustainable development roadmaps. The paper that he has in this special edition is called Vertical Farming Limitations and Potential Demonstrated by Back of the Envelope Calculations. So, Sam, I know you have some slides you want to share, so go for it.
Great, thanks very much. Let me pull those up. Is this working? Can you see my screen?
Yes, I can see them.
Wonderful. Okay, so I'll get started just jumping straight in. It's great to be here. Thanks very much for organizing this. I think it's a super exciting event. And let me begin by saying that we need new ways to produce food. We have a growing population. We have climate change that we need to adapt to. And we also have to minimize the mounting environmental burdens, which are in large part driven by our food production. And so we wanted to ask, how can vertical farming contribute to this? Vertical farming is where plants are grown indoors on shelves using artificial light to power plant growth. Now, because it's done indoors, you should be more resilient to external weather conditions. And because you're growing upwards, you should be using less land in theory. And this also means that you can grow plants closer to big cities and reduce transportation costs and so on. But the question is, can vertical farming actually compete on a large scale? Is it viable from a financial perspective? And this isn't super easy to ask or answer because vertical farming is still very much in its infancy. And there's not a lot of information reported by companies on what the cost of vertical farms are. And so we wanted to try and come up with an approach to get a sense of how viable would vertical farming be. And to do this, we wanted to start by just trying to estimate something simple. And this comes from the fact that in vertical farming, you need huge amounts of energy for powering plant growth. And so to perform a kind of sanity check to see, does vertical farming work, we just set out to estimate what's the cost of supplying this energy itself. And to do this, we looked at the cost for dry plant matter. So that's akin to something like producing wheat or rice. As staple crops, like wheat and rice, are the dominant source of our calories today, as well as the dominant source of the environmental impacts from plant-based products. And so should we want to reduce our pressures for tomorrow, we really need to be able to work on these kinds of scales. And so to break this problem down, we can first say, well, what's the energy that we need to end up with in plant matter? And that can be estimated by assuming plants are largely protein and carbohydrate and have a rough energy density of around 4 kilocalories per gram, or about 20 megajoules per kilo. We then need to know, okay, what's the efficiency of converting the energy that we're putting in into the biomass that we can end up harvesting? And we derive this based on reported values in the literature for how much energy you use, for what type of plant you end up harvesting, and you can find more about it in our paper, but we end up with this value of about 2% as the current best for vertical farms. So 2% of the energy you're putting in, you actually can get out in the plant matter that you're producing. And putting this together, you end up with this overall electrical energy demand of about 1,000 megajoules per kilo of dry weight. And combining this with the cost of electricity at the break-even cost, which is about 1 cent per megajoule, will end up giving you a total cost for producing 1 kilo of dry plant matter just from the electricity alone of $10 per kilo of dry weight. $10 per kilo. This simple sanity check shows that just from this electricity cost alone, without considering all the other nuances and complexities, vertical farming is at least 1 to 2 orders of magnitude, or 10 to 100 times more costly than the production of current staple crops. We can also do similar types of calculations for the environmental aspects of vertical farming, and you can see more in our paper about this, but we find limited environmental benefits there as well. The story's a little different for leafy vegetables and fruits, and you can find more about that in our paper, but ultimately we come to the conclusion that we're not really able on relevant timescales in the coming years to be able to produce staple crops in an affordable manner and offer environmental benefits. And so we don't think that vertical farming can substantially contribute to near-term global food solutions. It may work in some regions, in some places, but ultimately it's not the food of tomorrow on a large scale. And so with that, I think it's a great opportunity to thank very much Plant Physiology for this great journey, for Andrew for putting on and helping to organize this event. And I'm super excited to hear what the other speakers have to say and to have some good discussions. Thank you.
Thanks, Sam. That was exactly 5 minutes, so that's awesome too. And yeah, I encourage everyone to read Sam's paper. He goes into all of the different energy sources and does like cool calculations for each of them, so that way you can do your own back of the envelope calculations if you're interested also. So it's a great guide. So yeah, our next speaker is Claudia Vickers. Claudia is a professor at the University of Queensland of Technology and director of BioBuilt Solutions. Her research sits at the intersection of synthetic biology, metabolic engineering, and isoprenoid biosynthesis with a focus on rational strain design and pathway optimization in microbial and plant systems. Claudia previously led SISTRO, that's Australia's National Science Agency's Synthetic Biology Future Science Platform, and has played a central role in shaping national and international strategy in engineering biology, including co-chairing the World Economic Forum's Global Future Council on Synthetic Biology and advising a number of different international food organizations. Claudia's paper is still under revision, but is going to be Plant Agriculture and the Anthropocene, Improving the Carbon Footprint Through Synthetic Biology. So Claudia, go for it.
Thank you very much. It's a real pleasure to be here. I wanted to just second Jonathan's comments around the amazing leadership that Andrew has shown in this space, and the mentorship that he's provided all of us has been very, really extraordinary. And he, as well as the reviewers of our paper, which has just been accepted, thank you, pointed out the need for Fermi analysis, which I knew, but because I was running dreadfully late on submission, I had sort of left for the revisions. So if any of you online were the people who reviewed our manuscript, I thank you very much for pointing it out and pushing it a lot further and a lot more impactfully, I think, than it was originally. And I want to actually tell you a little story, because you all have the opportunity to read the manuscript, but I want to explain to you how we ended up writing this manuscript in the context of this this focus call. So I'm a plant molecular biologist by training and started in applied plant engineering and crop engineering, and segued into some more basic research, and then sort of after a decade or so, I actually moved into microbial metabolic engineering, because things were awfully slow in plants and it was a bit frustrating to get to impact. And having grown up on a farm, I've always been extremely impact orientated, and I wanted to see my science making a difference in the real world. And I thought, okay, well, I'll shift to microbes. And indeed, it is much faster in microbes. We can control things with a lot more finesse, specificity. The engineering goes a lot more quickly. In microbial systems, and the regulatory processes also can go a lot more quickly as well, because you're dealing with contained systems. And that was great for the last couple of decades or so, until the last, I guess, the period of time that Merritt mentioned. I was in CSIRO for 5 years, building a collaborative community of practice across Australia that the objective of which was to accelerate our ability to bring synthetic biology solutions into the real world. And it was an amazing and very unusual program where we co-funded 100+ fellowships and studentships, and really built the next generation of research leaders in the country focused on advanced engineering of biology and solutions in the real world. And we got about half a dozen or a dozen or so startup companies that came out of that, which was great and exciting over 5 years. But I thought we didn't really do very well. I thought we could have done a lot better out of that, 300 odd people who were involved in that program. And I realized at the end of 5 years in a government research organization that I really had no idea what it was like in the real world to take a technology from an idea and get it into the point where it's having impact. And I did something a bit weird. I actually jumped into startup companies, and I spent a couple of years as a CSO in startup companies. And I learned really hard and really fast what it's like to try to get technology in an environment where you don't have necessarily deep institutional pockets behind you. And I learned a whole lot of other stuff. It was sort of like a vertical uphill learning curve. And I came out of that with a much better understanding, at least of the startup side, and also a much better appreciation for, the things that sit behind company decisions and, pointy end R&D decisions. And then the year or so, a couple of years since I came out of startup companies, I actually have been running a consulting company, helping startup companies to work their way through, develop R&D programs and all that sort of stuff. And part of the reason I did that is because I wanted to give myself time to think very deeply and very purposefully about what I'm going to do in the next, the remainder of my career, 20 odd years or something like that. And the reason that I wanted to focus on that is because you can't think about big questions like that when you're running around trying to get grants as a university academic and just, trying to survive in a sort of fairly brutal career pathway that academia can be. And that gave me the opportunity to really consider what I wanted to do. And I wanted to address the most pressing problem that we face today, which is climate change. And I sat down and started thinking about what solutions my technology might be able to provide to the climate change problem. And at the time, of course, very focused on metabolic engineering and microbes. And I very quickly came to the sobering conclusion that there is very little, almost nothing we can do in microbes that can have an impact at the scale that it needs to have on climate change. And I looked around at what else is happening in biology in terms of scale, and agriculture is the obvious thing. So agriculture both contributes a lot to greenhouse gas emissions and has the potential, because it is massively scaled already, to sequester a lot of carbon dioxide. And so this manuscript really came out of what could we do? What sort of interventions could we have that could have meaningful effects at the scale required to make a difference? And it has to be, gigatons or in the order of magnitude of gigatons over a reasonable period of time. And so I sat down and sort of wrote out all the possible things that we could do to either increase sequestration potential or decrease emissions from agriculture, plant agriculture specifically. And of course, rice methane emissions, burning of stover and all these things came up as where the major emission sources come from. And the Haber-Bosch process is enormous in that space. So if we can replace 30% of the Haber-Bosch process, or really any reasonable percentage, that's the most meaningful impact that we can have at scale because of the emissions that come from that process. That was one of the findings that came out of this piece of work. But when we actually went and ran the Fermi analyses, and by the way, you can actually train generative AI to run Fermi analyses for you very successfully if you are careful and follow a number of caveats. And we mapped out how you do that in the supplementary materials and the things that you need to be careful for. But once I trained it and it had trained me on how to use it properly, it was extraordinary in terms of being able to rapidly and relatively easily be able to run the analyses that I wanted to run. So I highly recommend that. The thing that perhaps was most surprising for me, and even though it shouldn't have been, is that nothing matters unless it's at scale. So you cannot make an impact unless the process that you're trying to impact is happening at scale. And the scale needs to be at mega hectares of deployment. And those constraints really define the operational area of solutions that we can meaningfully implement in terms of impacting on climate change. I think that's probably a good place to stop. Hopefully I haven't spent too long there.
Awesome. Thank you, Claudia. All right. Up next is Jonathan. Jonathan is currently the science director at Rothamsted Research, where he joined in 2003. Jonathan is also an affiliated lecturer at the University of Cambridge and an honorary professor at the Institute of Aquaculture at the University of Stirling. Jonathan obtained his bachelor's degree in agricultural sciences from the University of Nottingham, followed by a PhD in plant biochemistry from King's College in London. Carried out a postdoc at the University of Cambridge, then taking up a position at Long Ashton Research Station in Bristol. So, Jonathan, please talk about your paper, which is titled How to Eat an Idea, a Roadmap for the Translation and Impact in Plant Biology.
Thanks very much. Yeah, it's me again. Really what I want to talk about here is the sort of specifics that, crystallized over a long period of time in my research, which was focused on trying to engineer plants to make omega-3 long-chain polyunsaturated fatty acids. We're trying to make omega-3 fish oils in plants for all the reasons that you could imagine why that's a good thing to do. The omega-3s are beneficial for human health. They're a diminishing natural resource. They're central to aquaculture, fish farming, all of these things. So there's a health benefit, sustainability story, all of these things. And economically, and we actually we didn't know we were doing Fermi calculations, but we were doing them a long time ago to discover that actually economically the case for trying to make engineer plants to make omega-3 fish oils made a huge amount of sense economically as well, because the cost of fish oils extracted from the ocean is probably an order of magnitude higher than the equivalent volume of a vegetable oil. So if you could engineer plants to make omega-3 fish oils, I'd always say this time next year we'll all be fish oil millionaires. Now, the fact that I've been saying that for nearly 2 decades indicates actually doing the translation was probably much harder than the metabolic engineering. And the metabolic engineering was hard because you have to put an entire pathway into plants to make it work. But we did that, and we regenerated transgenic plants that were making significant levels of our target fatty acids, EPA and DHA, but we did this in the lab, and then you suddenly realize that actually, exactly as Claudia alluded to, if you want to have impact at scale, you're going to have to use agriculture, and agriculture has been constructed over thousands of years specifically to deliver to the needs of mankind. That's the agriculture is the powerhouse that has allowed us to expand, because it provides energy and food for billions of people. So agriculture is the pre-existing infrastructure that you can harness to deliver all of these things. It's not like fermentation and other things where you have to scale everything up. The infrastructure for agriculture is already there. But actually, it turned out that we realized that although we got these plants in the greenhouse, we had no clue how to do the translation, how to do the scale up, how you would do that. And it's sort of that realization is why it is sort of crystallized in my little paper in this special issue. And I would encourage people to have a read of it, because it's sort of, I suspect it's something that is, our experience is something that lots of other people would have experienced or might experience in the future, which is that you find something really interesting and something with potential utility in the lab. But then you sort of suddenly think, well, how do I move this? How do I move this discovery into becoming a product? How could I eat this idea? And because actually it turns out that making a transgenic plant to do something sort of, funky is actually straightforward. And thousands of labs all over the world are doing this. And thousands of papers are getting published every year, every month, describing the fun thing that you've done. But actually, in terms of understanding how to do the translational side of it, things, then that sort of level of understanding sort of falls off a cliff a bit. There's many less papers telling you how to do the translation. And first, and one of the things that we encountered, so I completely chime with the paper that Merritt published about the importance of doing field trials to validate things. Absolutely critical. For us, the first thing was to do field trials. In Europe, doing GM field trials is still really, really difficult, really problematic. It's got easier in the UK, mainly because we've put a lot of effort into trying to just demonstrate that they should be the norm. But doing a field trial is really, to me, is just the very first baby step on your pathway to commercialization and translation. So this, field trials, absolutely critical. People need to think about doing field trial as a completely normal thing to do. They should build it into their experimental design. They should have that as the, as the sort of, the point where you can assess whether you've got a minimum viable product or not. You really need to test it. If it doesn't work in the field, it's not going to work in the real world. It's not going to be a thing that people will use because a farmer is not going to try and sort of nursemaid your little plant through the growing season. They just want to put it in the ground and grow it with a minimum amount of fuss. So we've my paper is really trying to describe about the sort of the journey that we went on, because we've been doing this for I would say it feels like we've been doing it for decades, because we have been doing it for decades, and so I would encourage people to read that. But this slide tells you about the journey. Even this is when we got into the field of understanding how you could use the product from the field, which is the oil, and the oil is used in aquaculture to feed fish. So that's why there's a picture of a salmon farm in the middle of that, and then how then the salmon would then get to market and be consumed by the consumer. And we're still on that journey, and it still takes a long, long time. And the reason we're on that long journey is because, as it says here, you realize there are a huge number of hurdles that, in academic research, you don't think about, like GM regulation, how you get through regulatory approval to grow at commercial scale and to bring a novel product through regulatory approval for feed and food use. Intellectual property and freedom to operate. Again, most people in academia, are blissfully unaware of intellectual property and whether or not they have FTO. But if you want to bring something to market, you have to think about these things. Business development, commercialization, all of these things, really critical. So I'm going to stop in a moment, but this is my, because I'm trying to be positive and trying to help people to think about this. Read my article, and I think I found doing the translation actually much more, it was more challenging, but it was more exciting than doing metabolic engineering. Metabolic engineering and synthetic biology sometimes to me feels like you're on a hamster wheel. You're just running that DBTL cycle round and round and round. And this is a way of getting off that DBTL cycle and actually getting to something that has a tangible output. But you need to have an entrepreneurial mindset to adopt that, and which is a little bit different from the sort of the academic mindset, but yeah. So I mean, the most important thing is take pride and pleasure in your achievements. This is a drone shot of one of our GM field trials here in the UK in Suffolk, and I sort of did a quick calculation. And there's something like about probably 1 million plants in that field, derived from one event, and that's something we made. We made that one plant, and and now we've got 1 million of them growing in the field, and that makes me feel really great. But also, it just immediately encapsulates that's the power of agriculture, and that's how agriculture can help get us out of the imminent looming crisis that we have relating to climate change and food security and all of those things. So yeah, so I'll stop there, and we can talk more in a bit, I think.
Right. Thanks, Jonathan. And I encourage everyone to read Jonathan's paper as well. It kind of talks through a lot of the trials and tribulations that are involved in taking a discovery to market. And like you mentioned, there's a lot more things about, the regulatory landscape, the funding issues that might come from trying to fund a project that's over 20 years long in the public sector. Along with like the logistics of seed movement, the freedom to operate, and yeah, finding commercial partners is there's a really good roadmap there to do that in that paper. So yeah, the last panelist here is Stephen Burgess. Stephen is an assistant professor at the University of Illinois Urbana-Champaign. Stephen's lab works on understanding the genetic regulation of photosynthesis with the goal of crop improvement. Stephen did an undergrad degree in biochemistry at the University of Edinburgh and obtained a PhD in molecular biology at Imperial College in London in 2011 before joining the plant biology department in 2022. And Stephen's paper is How Synthetic Biology Can Realistically Impact Crop Improvement in Agriculture.
Thanks, Merritt. So I think all the previous speakers it's been kind of a great lead into what I'm going to talk about. I would say that, so I'm co-author on this paper. It was actually Merritt is this first author, but there are also Andrew and Catalin and a number of other people on this. So what this really came out of was, I think this was the conference that Jonathan alluded to earlier in the discussion, which was a workshop organized by Andrew and Catalin at the University of Florida, which was trying to address this idea of like, how can we realistically use synthetic biology to make improvements in the world? So I thought it's actually really interesting to hear Claudia's story, and I think that it's one that I think is actually really inspiring that you can stop at some point and say, what I'm doing now is probably not the most useful thing, and I think that is something that people need to do a lot more. So from my own personal view, I'm a molecular biologist by training. I know that we have a worldwide audience and everyone is in very different systems, but in the UK, the places I went to, I was very much like a bench scientist, so although we talk about, the problems that we meant to solve and what's not, normally actually it's fairly disconnected from what's actually going on in the real world. So as many synthetic biologists or other molecular biologists often, it's kind of by second hand, or like, I'm sure they feel this kind of disconnect of like, how can we make a real impact? And I think this was, I had some discussions with Andrew about similar topics of the future. And I think that one of the things that he was concerned about was like, the kind of the health of plant science as general, and like what as early career researchers, like being, what's the word to say, kind of maybe slightly misguided in what they think they can achieve, or overselling what they're doing, or kind of, I guess it kind of feeds into the whole idea of this issue, which is like proposing solutions to things which are completely, unrealistic. So I think that was kind of one of the main aims of this workshop, was to try and bring some realism and try and work forward, like how we as a community can start to move forward and kind of address this. So there were kind of three main things that came out of this. This was actually part of the breakout sessions during the conference, but this was where are we now as a field in synthetic biology? Where should we be? And how do we get there? I think those are the things that we were trying to address there. So, and actually much of this stuff has been said already, but I think kind of the key additional things which kind of brought out here really is this. We have this disconnect, right, between lab scientists or synthetic biologists and those working in agricultural science. So often they're completely different communities, very rarely meet. And I think this is actually a really big problem when lots of the challenges that synthetic biologists are trying to address are really those in agriculture. So if you don't have a very good understanding of the problems themselves, then you end up making the wrong type of suggestions, I think is what it was. So a number of things that came out for this is like, what other problems do we have? I mean, we've talked about this already, but it's this idea of the value proposition. So is what synthetic biology or whatever technology you're developing, is it better than what already exists? Like, can it actually be more cost effective? So it may be a cool solution, but is it actually worthwhile doing? I think that was one of the things we were trying to address, and I think this idea of Fermi calculations is one way of getting at this. This is particularly important for the ag industry, which is deeply conservative and it requires hard returns on the money, right? So although you can do something, doesn't necessarily mean it's going to be the best solution. Other things that came out of this was this pressure to hype the work. And this comes from all levels of the research infrastructure, right the way from funders, press officers, and scientists. We live in the attention economy, and there's really this demand to try and get your work seen. So there's often people extrapolating from very small sample sizes or small number of experiments to saying that this is going to be a very large solution. And so there was kind of an idea that we should get away from that. I'm conscious of the amount of time that we have here. So, where we should be, I think much of it follows on from where we are now. It's kind of almost the opposite. But I think the key things are, with this idea is trying to find more spaces where you get this interaction, this increase in interaction between agricultural scientists and those working in more kind of the molecular sciences, and kind of working together to jointly identify good use cases and value propositions. So rather than suggesting solutions, maybe actually working together to work out what's going on. New ways of finding out how we can fail quickly. So Jonathan mentioned there's a very long time scale. Is there a way that we can get to understanding which things should not be pursued as soon as possible? And again, the Fermi calculation is one way of coming into. How do we get there? Well, I think there was a big emphasis on this idea of one is more meeting spaces, but also education. So other new ways, if we're training the next generation of researchers, can we have things such as cross-disciplinary training internships or educational, kind of interdisciplinary conferences, like organizing more things where you get people from different disciplines coming together. Because we have this people often now are so specialized, often don't kind of get outside their own field. But finally, again, it's like one of the things that was brought up, and often one of the kind of complaints people sometimes make when they get to this is that as a lab scientist, I have no idea how to run a field trial, right? And it seems very overwhelming, and kind of it can sometimes seem very impractical. It's very expensive, and it's often out of reach of people. But is there a way that we can work to kind of bridge this gap? So one solution that was suggested with this idea of like university help centers. Again, it varies across the world, but particularly in the US, we have the land grant system in the Midwest, for example. And these often have departments, like if you have like crop science departments with experts who really do this type of work routinely. And it's really trying to start to bridge that gap between the kind of lab scientists and those in the field to try to actually do proper rigorous work that can be reproduced. I'll stop there to try and leave some time for questions. Yeah.
Awesome. Thank you, Stephen. And yeah, there's a number of great questions in the chat. So I'm going to start off with a couple of questions that we had pre-submitted. And so this one is more for Stephen and Jonathan, but the other panelists are also welcome to answer it. But the question is, if you could change one thing about plant science training or culture to improve numeracy, realism, and relevance, what would it be?
I'll have a go at this. I think, I sort of alluded to it when I talked about having an entrepreneurial mindset, but just sort of being able to understand the, and Stephen actually articulated really well, the sort of the relevance of a technology. Just because you've found something cool doesn't mean that it has utility or even, potential to be a product. I sort of despair, but maybe it's just because I'm an old person, I go to meetings and people say, oh, wouldn't it be cool to, and then say, some completely, implausible thing. And it's like, why do you want to do that? I think we should be challenge led. I think, and sadly, the world is facing an enormous number of challenges. So, we should be dealing with the challenges, of trying to, mitigate climate change, deliver food security, better nutrition, all of those things. But, it needs to be based in realism. And that's that, what can I really do? And just having a sense of realism and saying, what is it that I can bring to the party that's not completely overblown in claims?
Yeah, I mean, I think it's a hard one to answer. I mean, probably the thing that to me seems a most practical solution to this is this slightly changing the way like our undergraduates are taught. So like reducing over specialization and trying to have more, even if it's short courses within degree programs, where you get, and this was also mentioned actually in the opinion piece, with this idea that at least if you're doing a molecular degree, maybe you should have at least one semester of like kind of intro to crop science or something for like molecular students, and vice versa. Like I think these kind of ways of being more grounded in a wider range of disciplines. But yeah, I mean, I think it's a hard question to answer.
Kind of gets a little bit at that like depth versus breadth of education and what is right. And yeah, there is value in going out to different fields and learning different things, but there's also value in collaborators that do have that breadth of knowledge, or that depth of knowledge, sorry, that can, collaborate on research projects. And often that's incredibly valuable as well. The next question is more for Sam and Claudia, but we've heard a lot of caveats and concerns about overselling. Where do you think a lot of this overselling comes from?
I'm happy to dive right into this since my mic's already off. It comes from a lot of different areas. So in academia, not only are we able to make some pretty unrealistic claims about what our science can do, we're really encouraged to claim far and beyond without any serious evidence around what the potential impacts of our often very basic science might be. So there's a and as we've already discussed, there's a bit of a lack of realism. And then there's the startup community, which has eaten the entire Gartner hype curve around emerging technologies. So one of the most traumatizing meetings you can go to is a startup community meeting talking about their great ideas to a bunch of investors. It is the most painful experience you will ever have. It can only be managed with copious amounts of alcohol, which also seems to be. So it's this kind of financial drive. What's the best story you can tell to get the best investment funding? And one thing I really learned working in parallel to the startup community is that there can often be quite a massive disconnect between the science and the claim and then the funding gap. And I mean, I'm will put my hand up and guiltily say that when I first got into synthetic biology, I really was a very strong proponent for it because I wanted to do this thing that didn't really exist in Australia, and I needed everybody to understand what the thing was and what the value was. And I even actually ended up in the cabinet room explaining to the Prime Minister what synthetic biology was. That was Turnbull at the time. In the back of my head also going, this is insane. And that was a process that we went through to actually get the funding approved to get our Australian Council of Learned Academies to do a road mapping horizon scanning report on this emerging technology. And that's how things happen. So you have an emerging technology, all this excitement happens, and people suddenly go, oh, we might be able to get some grant funding out of that. Awesome startup funding, and everybody piles on. And this technology in particular takes a lot of time, as Jonathan has already mentioned. It takes a lot of time, even in microbes, but definitely in plants. And so the Gartner hype cycle has been really extended. So you have this innovation trigger, you have this peak of inflated expectations, you have a trough of disillusionment, you have a slope of enlightenment, and then you have ideally a whatever it is, a plateau of productivity. And we've actually hit the trough of disillusionment hardcore in microbial engineering in the last sort of 24 months. You've seen a whole lot of companies go under actually, or retract, or file for bankruptcy, Chapter 11. And what that means for us right now actually is very exciting for me, because although there's lots of bad news in the news about companies that have been working for a decade or 2 decades to try and get technology off the ground, it also means that we're moving on to the slope of enlightenment. And that means that we can realistically start making predictions about what we can and what we cannot achieve. And instead of saying, what can we do with this technology, we can say, what should we do with this technology? And I think that that's the essence of this entire special issue, in fact.
I think I'll echo as well many of the things that Claudia just said. And I think some of the things that's also important for us to consider as to how we can actually get away with some of the claims that we do here is we can look to other fields like the climate change field, where when they run their models, they have standardized pathways and standardized ways of doing their modeling approaches. And I think that's somewhat of a more unique approach, because developing standards is a lot of effort and takes a lot of time, but ultimately it's very much worth the effort of going to, because you can very quickly understand what scenario it is people are projecting and what that means in terms of the outcomes that they get. And I think that's not something that's necessarily translated so well to other fields of, we don't necessarily have the same standardized assumptions, and that doesn't then make it easy to understand what is it someone is trying to model and what that outcome is. Two people can try to come up with the same number and end up with very different conclusions, and that's not always clear as to how that happened, and what one person might be saying is this is kind of a best case and someone is saying this is a realistic case, and that can get lost along the way, because a lot of the time the models that we can end up using are quite complicated with many layers of information and detail, and understanding exactly what the assumptions that go into that are can be challenging to figure out. What does this mean? Is it optimistic? Is it realistic? Is it pessimistic? And so I think being clearer on that front and also being more transparent. And I think this is also where back of the envelope or Fermi type calculations can really help, where it's immediately obvious because you're trying to simplify the question and the answer down into a simple equation or simple form. It's obvious as to what's happening, and that can make it a lot easier to communicate how realistic is what you're doing and what its actual outlook is.
Can I add this? Yeah, so I mean, I think this is something I've thought about a lot too, and maybe this is slightly unrealistic, but one thing that I would like to try and encourage everyone to some extent is to perhaps return to the idea that science is about disproving hypotheses, like putting them forward. I think one of the problems a lot is that when, as a researcher, and particularly if you're invested in it, you want to see it succeed, right? It's like, oh, I'm trying to put this product out, and then it's like, or I'm working on this technology, and perhaps a somewhat sanity check on it is like, I believe in this, but like let's try and be rigorous, trying to show as like the best I possible can that is this actually going to fail, and like and try to show would it fail or not. I mean, it's the idea of like you put forward the hypothesis and you design an experiment to try and disprove it. I'd love to actually talk to people in the physics community about this type of thing. You think about the idea of like CERN, and and they have these hypotheses or like theories about the way that certain physics works, and they built this giant machine to try and disprove it, or like you're going to slowly, rigorously go through each one of these steps to try and work it out. And I don't know how much that's possible, but it's like when you're trying to put forward a solution, trying to think, well, if this was someone who was like, I'm trying to convince someone, really, if you work the best you can to show that it doesn't work and it still does, then you can really be on a good way of trying to convince someone that it's actually worthwhile pursuing.
Yeah, absolutely. And Claudia, you're unmuted, go for it.
I just wanted to add a thought on that. I just did a quick ChatGPT question in the background here. The average cost of bringing a GM trait to market is $115 million US. And that means that the trait has to be worth much more than that, ideally tenfold more, to make it worthwhile for a company or investors to invest in that. That's one side of it, but it has to be taken up as well. So for the farmer, the trait has to save them money. It has to be worth buying that trait, and it has to cost less money than it saves them. And the profit margins are tiny. So you really have to do a very fine, well-educated cost-benefit analysis on whether it's worth pursuing. And you need deep pockets and patient capital for this kind of technology.
A good point to bring up. I'm going to go, we have 5 minutes left, so hopefully we can answer these next couple of questions kind of briefly. But the one that I want to have you folks answer is from Sarah, and it says, as a fundamental scientist, I always struggle with the impact part of proposals. It's very hard to be fully realistic when it comes to seek funding because I know that success will depend on the impact section. On the other hand, I'm also reviewing other people's proposals and I can feel the same struggle. So the feeling that we're all guilty in perpetuating the current funding system. Are there any ideas on how we can break this circle and to make a much more honest system in how we request for grant funds?
I mean, I think it's a real problem, and Andrew and I discussed this recently. I think you look at journals like PNS where they have an impact. I can't remember what it was, a significant statement, which is sort of like a license to print money, basically. I think it's not helpful. The intention is entirely good, but the outcome is entirely not what was intended. And so, in the context of funding, I think we have to sort of try and grasp the nettle and actually suggest to our funders and policymakers that there needs to be a degree, some real realism in terms of when this impact will be manifest, and because there's no point in asking people who don't know how long something will take when their impact will be manifest, because they'll invariably say, oh, in 3 years' time, at the end of my grant, we'll all be up to our knees in £10 notes, and my unicorn farm will be in full swing. It's not helpful. So we need to get the policymaker, the funders, to reconfigure their thinking. That's maybe that's on a par with running a unicorn farm, but I mean.
Yeah, I'll follow on from that. I think it is ridiculous to have such a strong emphasis on impact for basic research. Nobody can pick the winner at the basic research stage. Absolutely nobody. They shouldn't be asking that question, in my opinion, for basic research funding. For applied research funding, for translational research funding, it's very different. In academia, academia, I think, is not the place, by and large, where you will be making impact, unless you're in a subset of unusual academic environments like Rothamsted. CSIRO is not an academic organisation, it's a very translational organisation. But by and large, most universities outside your MITs and such forth, they don't exist to have impact in the real world. They're called ivory towers for a reason. They exist for the training of the next generation and for exploration of science and the natural world around us. There is a push at the moment to drive universities to change their business models because the current business models are failing. But that's a different discussion. In 5 and 10 years, you'll see very different questions, I think, being asked, and funding structures are being changed all around the world, not just in places where the order of things seems to be falling apart anyway. So if you really want to have impact in the real world, you probably want to get out of a university.
Stephen or Sam, is there anything you want to add really quickly?
I think it's just recognizing that there is a place for basic science, and you don't have to always extrapolate from basic science saying you're going to have an impact. Like sometimes the impact is knowledge, right? But I don't, how do we address that? I don't know. Maybe it's a lot of this stuff has to be bottom up. So I guess if you're on funding, if you're reviewing people's grants, if you don't see them extrapolating, yeah, it's great. They don't make those type of comments like, oh, this doesn't have any impact, right? Because I don't know the way to address that other than working from the bottom up.
I'll also just add on to the back of all of this. I think there's a challenge in understanding when we're trying to communicate what the impacts mean as to what the underlying numbers represent. And I think that can drive in part overselling or miscommunicating what it is you're trying to do. For example, when it comes to, say, nitrogen use in Haber-Bosch, it's a couple of % of global energy use and a couple of % of greenhouse gas emissions. And that might sound, in those terms, not like a huge deal in that we still have 90-something% of the problem left. But a couple of percent of an absolutely huge problem is still a big problem. We have to get more comfortable with understanding the different scales and what the numbers really mean. And so I think having kind of a more consolidated set of, here are the key numbers you need to understand the scale of the challenges and what it is we can do can really help in terms of communicating and avoiding, when it comes to such impact sections, having to feel like you have to oversell, and actually pitching it at the right level, I think would be a helpful step forward.
Thank you all. I know we're at 8, so I know Mary might have some final words to close us off, but I want to say thank you to all of the panelists for their answers today and their honesty. And I encourage all of you to read their articles as they're really insightful. This is a really great special edition. So with that, Mary, I'll pass it off to you.
Great. Thank you, Mary. You did a wonderful job. And, what timing, my God. Brilliant. Claudia, Sam, Jonathan, Stephen, really enjoyed this tremendously. Once again, thank you, Andrew, for pulling this all together, pushing the envelope in how we think about things. I think I've known you a long time, and you've always been somebody who's pushed people, and we really appreciate that nudge. Thanks, everybody. I really enjoyed this. Fantastic. See you all next time. Bye-bye.